Okay. Okay, is the screen visible, please?
Yes, please.
Okay, and can everyone hear me, please?
Yes, I can. So I guess everybody should be able to hear you.
Okay, thank you. Good morning, everyone. It's a pleasure being in your midst. I'm the one who is honored to be here. My name is Tosin Afowowe, and what these mean, is the pointer visible, please?
Yes, it is.
Okay, so, and what this means is that we are all learners. I'm a learner like every one of us, so let's learn together. So this morning is just a brief talk on basic molecular biology, genetic materials, and the replication process. Just an introduction. Okay, thank you, Dr. Francesca. So, definitions. Simply put, as we say from high school, biology is the study of life. And if we take it a little bit further, we can say that molecular biology can be just simply defined as a study of life at the molecular or the fundamental level. At this point, I would like to ask if anyone would like to give a definition of molecular biology, perhaps from literature you've seen or from your own experience. Does anyone want to give a definition of what he or she understands as molecular biology? Because this is simple. Anyone, please.
Yeah, you can raise your hands. You can send it to your chat. Dr. Francisca, are you trying?
Study of life in the simplest form. Okay, thank you very much. Thank you. Okay, moving on. So yes, that's the most simple form we can take a look at molecular biology. However, if we go a little bit more complex, we can say it's the study of the structure, function, and composition of the molecular components of life. And it's associated with a couple of other fields like the biochemistry, genetics, and cell biology. And its main focus is on how DNA, RNA, and protein synthesis interact, and now they, they regulate each other. Okay, thank you very much. Is the understanding of interactions between various systems of a cell. Thank you. So we can also describe them this way, as literature shows us. So one thing we should bear in mind when we talk about molecular biology is the central dogma. This central dogma was first developed by, Francis Crick in 1958. This central dogma is simply a theory that says that genetic information flows only in one direction, from DNA to RNA, and from RNA to protein. So a unidirectional flow of information, or from RNA directly to protein. However, as we know, things evolve, science keeps evolving, and there has been some counter reactions to this definition of the central dogma. So some scientists have developed over time exceptions to this theory. And one of them is the concept is the prions, which are infectious proteins. And I believe prions is important in vet medicine. And they say they replicate throughout their replication does not go through a DNA or an RNA intermediate. This surely goes against what the central dogma is telling us. If you also look at some viruses, their replication has shown that genetic information does not flow in just one direction. You could have it moving back and forth in between DNA and RNA to finally get their complete replication cycle. So as far as central dogma is central, there are also exceptions to this rule. A look at this picture shows us what the central dogma is. DNA to RNA to protein, a unidirectional flow of information. It also tells us that DNA replicates within itself. That is the experiment replicates to make more copies. That is expected. And DNA to RNA is transcription facilitated by RNA polymerase. And RNA to make protein, the process is called translation. And the ribosome is very important with this. If we take a look to the left, to the right, we can see the same picture, but with some exceptions. That is, from RNA, now we can have DNA, which is reverse transcription. And we can have replication within RNA, negative sense to make positive sense to make, negative sense to make positive sense before you have proteins. So these two are exceptions to the central dogma, showing them, giving us an illustration of what these exceptions look like. This is the human cell from way back in biology, and this is very important to molecular biology. I say molecular, the fundamental cell is a fundamental part of life. And then we have nucleus, the nucleolus. A lot of activities take place here, and in the cytoplasm as well. As we know, DNA is present within the nucleus. RNA exerts its impact in the cytoplasm, and these are the ribosomes, very important for protein synthesis. So we should bear in mind that most of what we are doing takes place in the human cell and in the cellular component or counterpart of other organisms as well. A look at genetic materials. Genetic materials can be defined as the hereditary substance within the cell. In the cell, hereditary, know what hereditary means, parents making offsprings. So this genetic materials carries all the information specific to an organism. And depending on the organism, it could be DNA, RNA, or perhaps something else. But we as human beings, we the eukaryotes, DNA is our main genetic material. For some viruses, it is RNA. So everything that an organism will be is coded for or determined by the DNA. And as I said, it's either DNA or RNA, depending on the organism. And some scientists also classify genes as genetic materials, while proteins are not classified as genetic materials because they lack the ability to replicate, which is a very important feature of what a genetic material is. As we've shown in previous diagrams, DNA replicates and RNA can also replicate as well. So proteins, they do not, and they are not genetic materials. In eukaryotes, plants and animals, the DNA is mainly located in the nucleus, and that's nuclear DNA. Mitochondria can also have DNA as well, but these are to a lesser extent. You can say we have mitochondrial DNA. And DNA is a molecule that carries genetic information for the development and functioning of an organism. Like I said, anything an organism will be is controlled by the DNA. If the organism is going to be tall or short, if it's going to be yellow or dark, mixed color, how the face looks, the eyes look, everything is determined by the DNA. And DNA was first identified by a Swiss chemist called Johann in the 1860s, as far back as the 1860s. He had the idea, he knew there was something that looked like DNA. He called it nuclein back then. Later on, it was called nucleic acid. But he didn't know what it really was. He couldn't find the structure. A look at history tells us that the structure was finally discovered by two British scientists in 1953, James Watson and Francis Crick. But as we'll be looking at in the next couple of slides, these doesn't tell you the complete story. The structure of DNA, which I believe most of us, or maybe all of us, have seen at one point in time, it's a long double helix shaped structure. It has a sugar phosphate backbone on the outside, and its bases are on the inside. That is what the structure looks like. It's double helix. I'll show us a picture very soon. Sugar phosphate backbone, combination of sugar and the phosphate group, and the bases, the nuclear bases in the inside. The two strands run in opposite direction, they're anti-parallel to each other, and the double helix is stabilized by hydrogen bonds between the bases. However, these hydrogen bonds are relatively weak. So the sugar here is the deoxyribose sugar, and the bases, they are bases, or you can call them nucleotides. They are 4 in number: adenine, cytosine, guanine, and thymine. Now, a look at DNA. So this is the structure, a double helix, the sugar phosphate on the backbone on the outside, and the nuclear bases on the inside. So there is a rule for binding. Adenine binds only to thymine. You can see yellow, red. Cytosine binds only to guanine, blue, green. So the adenine and guanine, they are called purines, cytosine, thymine, called pyrimidines. So this is the structure of DNA, the popular double helix structure. Now, like I said, a good number of literature will tell us that it was discovered by this structure was discovered by these two smiling guys called James Watson and Francis Crick. But as I said, this is not the full story. 2 other people are important to this discovery, maybe even more important than these guys. A major player is Rosalind Franklin, who is also British, and Maurice Wilkins, who is from New Zealand. So the notes of this lady, the previous experiments that this lady did, facilitated the discovery of this structure by these 2 guys, but they take most of the credit. And in 1962, these 3 were recognized, awarded the Nobel Prize, but unfortunately, she had died by then from ovarian cancer, perhaps due to the kind of experiments she was doing and due to lack of effective biosafety procedures. So she couldn't be recognized posthumously for a Nobel Prize. So when we talk about DNA, the structure, the discovery, we shouldn't think only of these 2 guys, but also consider the sacrifice or the immense amount of work put in by this lady and this guy as well, irrespective of what literature tells us. RNA, which is another genetic material, it's a nucleic acid very similar to DNA, but with a couple of differences. First of all, it's only has a single helical strand. DNA is a double helix. Secondly, the sugar is a ribose sugar rather than a deoxyribose sugar. And thirdly, the bases are the same, nucleotides are the same, except for thymine. In RNA, we don't have thymine, we have uracil, and it's the genetic material of some viruses. There are 3 main types of RNA. Each has its own function: messenger, transfer, and ribosomal RNA. mRNA acts as the messenger. It takes the instructions from DNA to ribosomes where proteins are made. Transfer RNA picks up specific amino acids from cytoplasm, takes them to the ribosome for the protein synthesis, and ribosomal RNA makes up about 50% of the ribosomes found in the cytoplasm. And we know that that's a very important organelle for protein synthesis. Just the structure of RNA, single helix, the bases, uracil here. So instead of thymine, adenine binds to uracil, cytosine binds to guanine with hydrogen bonds, and the structure of the messenger, ribosomal, and transfer RNA. Talking about DNA replication, the process is semi-conservative. As we said before, DNA replication means DNA making copies of itself. Actually, this process takes place before the cell divides, and it's very important for our growth and our reproduction. I'll be running down the procedures in DNA replication, but we should just know that basically it's just DNA making copies of itself to ensure that life continues. Firstly, an enzyme called helicase opens up the double strand of DNA. That double helicase opens it up. Now, these separated strands, now two in number, as we know, they serve as templates for making new DNA strands. And DNA polymerase is important for making the new strands, but it doesn't know where to start. So a primer, just a short piece of RNA provided by an enzyme called primase, tells DNA polymerase where to start DNA synthesis. So DNA polymerase binds to this primer and starts adding new complementary nucleotides to form a new strand. Another enzyme called exonuclease cuts away the primers once the base matching is completed. And then, to ensure that there are no mistakes, DNA polymerase proofreads the nucleotide it has added to form the new strand and form the new double helix DNA. When this is done, DNA, any spaces in between DNA, I'll show us why there are spaces in the next slide. DNA ligase seals up any space between the sequence of DNA, and then we have two continuous double strands. And then when the new DNA is formed, it automatically winds up to form a double helix. So that's how DNA replicates, and it's called semi-conservative because in the new strand, you have one strand from the old strand and a new strand. So semi-conservative, it's as part of the old strand, semi-conservation. These two pictures just tell us about DNA, a little bit more about DNA replication, and I'll try to use this to explain to us. This is the original strand, original DNA. So like I said, helicase comes somewhere here and moves this way from right to left. As it moves, it unzips the DNA. It starts loosening up the double strand. This side area, region, can be called a replication fork. Now, when the two strands are made, the two new strands are made. One is called the leading strand, and one is called the lagging strand. It's called the leading, this one is called the leading strand. This one on the bottom, this labeling shouldn't confuse us. It's on the one on the bottom, it refers to the new strand being formed. It is continuous. That's why it's called the leading strand. And it's continuous because DNA polymerase can only copy 5' to 3'. So this is straightforward. It is continuous. It is running towards the replication fork. However, the lagging strand runs away from the replication fork from 5' to 3'. If this is a little bit complicated to understand, I'll try as much as possible to explain, and with primary studies, we can understand better. But this is discontinuous because it is formed in bits and pieces called Okazaki fragments from 5' to 3'. And at the end of the day, RNA, this space in between, a ligase joins them up at the end of the day to form the double strand. So this is called lagging strand because it is not continuous. It is formed in little pieces called Okazaki fragments. This just talks, this is just about the same thing. I'll be rounding up with applications of molecular biology. We use them in research, of course. Any study, undergraduate, master's, PhD, whatever study, postdoc, molecular biology is very important. Also for disease diagnosis in laboratories, very important. Diagnosis of almost any disease you can think of, PCRs, primers for it. Then paternity test, which I believe is we are very conversant with. Paternity test is like an in-thing or a very hot topic in our environment. Forensic analysis, gene therapy, and drug design. Like you can take a look at a gene to design a drug, or we can start designing drugs specific to individuals rather than making generic drugs. We can design drugs for a specific group of people or for a particular person based on the person's genetic content. So these are my references. I want to believe these slides will still get to you later on, and you can have more study looking at these references. Thank you very much for listening to me and for having me with you.
Okay, thank you very much, Dr. Tosin. This is a period for question and answer. So if you have a question, you can raise up your hands, or you can send your question into the chat. But while doing that, I think I would like to also appoint somebody to represent the class. I'm trying to look for somebody that I can I can choose to represent the class. I don't know. Somebody should volunteer, please. So if you want to represent the class, you can volunteer. Just raise up your hand. Not for question now, but to represent the class. I've been choosing one person in all the classes. So I want to choose somebody here, but because there are a lot of people here, so I just want somebody to volunteer. Okay, somebody said I. Who is this? Festus. Okay, Festus is this. If there is no other person that is interested, so Festus, please, I will send you the attendance for everybody that's supposed to be here, and then I'm also going to send this lecture note to you, so you can create a group and share it with everybody in that group, so that they can have access to it while we prepare. So next week we are going to PCR. So I would advise you read ahead. PCR is polymerase chain reaction. That's what we are going to be doing next week. So you can read ahead and the application of that PCR. You can hear that. So, but why we are here today, you have listened to molecular biology. I don't know, it may be new to some of us, and it may be just a refresher to some of us. So please, if you have any questions, you can please put it on the chat, or you can also raise your hands, and you can, okay, somebody is, and then you can raise your hand, or you can ask questions by sending it to the chat. Because of our time, I would want us to do that in time so that we can be able to go ahead.